Hello everyone, this study is about accurate and proper simulation of a human intervertebral disc and particularly the annulus fibrosus. This study has recently been published in Acta Biomaterialia, and for more details please check out our paper. You can find more details and more results in the paper. This is the schematic of a human intervertebral disc. In the middle we have nucleus pulposus, and on the outer rim we have annulus fibrosus. If you look at it more closely and look at two collagenous layers, on one layer collagen fibers are oriented almost uniformly in one direction, but in the adjacent layer the direction of the fibers is in the opposite direction. So the orientation of collagen fibers changes in each lamella, and the direction of collagen fibers in each lamella is totally different from the nearby layers. However, almost all of the models homogenize two adjacent layers into a single layer with two groups of families of fibers. This is a very common and popular modeling technique, and it has been used um, since 1977 until now, and many people are using it. And one of the objectives of this study is to evaluate the validity of this modeling technique. Also, in between two collagenous layers, there is another fibrous network, which is called ILM, or Intra-Lamellar Matrix, which is mostly made of elastic fibers. And these fibers has an organized structure. Recently, Tavakkoli and Costi has measured the orientation of these fibers, and by using their SEM measurements, we could come up with this probability distribution for elastic fibers in ILM. However, no study have investigated the role of these elastic fibers on the responses of annulus fibrosus. Also, to validate the models, most of the people use a single loading condition. For example, they calibrate or validate their model in a uniaxial test or biaxial test. They do not use all of these tests combined. So we do not know what's the performance of existing models in all of these tissue level tests. Also, most of the people use idealized boundary condition and idealized motions to calibrate their model. So the objective of our study is to develop anatomically accurate constitutive models of annulus fibrosus by incorporating layer-wise structure of annulus fibrosus and by incorporating interlaminar matrix. And finally, we want to validate and evaluate the performance of these models under different tissue level tests like uniaxial and biaxial tests. Initially, we developed a micromechanical model. The micromechanical model takes account of the layer-wise structure of collagen networks and the collagen fibers has nonlinear stress-strain response with a content of 15%. And also, it takes account of elastic fibers, which are in the interlaminar matrix. The elastic fibers has the modulus of 1 megapascal. This 1 megapascal comes from micromechanical tests of single elastic fibers, and the content of elastic fibers was set at 10%. The fibers were simulated as truss elements, and they were embedded in a Hookean matrix. So far, we have developed the micromechanical model. However, the micromechanical model has one major shortcoming. Its computational cost is huge. Alternatively, we developed two hyperelastic models, which are much faster. In the first model, we have the contribution of the matrix as an incompressible matrix, and it's a function of the first invariant. You can find the definition of these invariants on the right side, and also the contribution of the fibers as well as the interaction between the fibers and the matrix. Go has this nice paper, and in their paper they make a very solid argument why constitutive models of annulus fibrosus should consider the interaction between the fibers and the matrix. Basically, they say the matrix should stiffen when fibers are extended. Here, we propose this simple yet accurate term to consider these interaction between the fibers and the matrix. We additionally use another hyperelastic model which has the contribution of the matrix as nearly incompressible Neo-Hookean solid and the contribution of the fibers, which is a little bit more complex. And this is microsphere or microplane model. You may find further details about this model in the Kanner's paper. In short, at each integration point in the finite element method, we evaluated this integral on the surface of the unique sphere by using Lebesgue's quadrature rule to evaluate the contribution of the fibers on the stress of the material. We evaluated the performance of our model in different tissue level experiments. This test is uniaxial test along the fiber directions, and the shaded area shows the experimental range, and all the three models were within the experimental range, and they had good agreement with existing measurements. This is also another uniaxial test, but this is along the circumferential direction. The orientation of the fiber is different from the previous test, and the blue points shows the experiment, and we had a very good agreement with measurements. This test particularly is very important because they performed both uniaxial and biaxial tests. So also we had a very good agreement with the in the biaxial test, and the shaded area, the blue area shows the standard deviation, plus minus the standard deviation of the test, but the blue circles shows single specimen. We found an interesting point when simulating all those tissue level tests. We found that sample shape and geometry substantially affects both uniaxial and biaxial stress-strain responses. For example, you can see that the stress-strain response of the hourglass shape sample is quite different from the cross-shape sample. So when someone wants to develop or calibrate his or her model in tissue level tests, he or she should consider all the geometric details of the experiments. For example, Bass didn't report the width of his sample in the axial direction. So you can see that how these variations in the geometry can affect the stress strain response. When simulating uniaxial tests by using anatomically accurate model versus the homogenized model, and this homogenized model is the conventional and popular method of simulating annulus fibrosis, we found that the strain field and the stress field is quite different between these two approaches. Although the gross stress strain response is nearly similar, but the strain field is quite different. You can see that the patterns is entirely different in these two approaches. Additionally, when we evaluated the response of the whole disk model by using layer-wise and homogenized approaches, we found that the compression and displacement responses of these two approaches are nearly similar. However, the stress field is quite different between these two approaches. You can see that the pattern and the magnitudes of the stresses are quite different in these two approaches. In summary, we developed three different models, one micromechanical and two hyperelastic models, which had a good agreement with tissue level and whole disk experiments. We found that variations in sample geometry substantially affects the stress-strain responses of tissue-level tests, and this highlights the importance of accurately simulating experimental conditions. And we found that proper simulation of individual lamellae as distinct layer are crucial to have a good estimation or accurate estimation of stress and strain fields. At the end, I should thank funding agencies and other co-authors, and thank you for your attention.